All right. Hi, everybody. I've got a few general administrative announcements for you, and then we can get into the material. So in terms of general administrative stuff, easiest one is that you should remember that there is an inquisitive due on Wednesday with questions about the T-cell receptor and T-cell development. So be aware of that. Another kind of general thing to be aware of is that on Friday of this week, we'll be having class on Zoom. I will email you guys the Zoom link, but just know that we're going to have Zoom class on Friday. If you'd like to be here, you can. If you'd like to be somewhere else, you can too. And on Wednesday, I'll tell you a little bit more about the plan for lab. There's one thing I'm kind of waiting on before finalizing all of it, so I'll tell you about that on Wednesday. Today I'm going to be talking about a few more pieces of T-cell development that we didn't see last time, as well as starting our discussion of T-cell trafficking. I will very likely be finishing T-cell trafficking on Wednesday, and I am very excited about that because T-cell trafficking would be a really bad one to do on Zoom because you wouldn't get to see me do interpretive dance. So I'm glad that we're doing T-cell trafficking in person. Last time we went through the steps of T-cell development, we saw the double negative T cells in the thymus, which can be divided up into 4 subpopulations, DN1 through DN4. We saw DN1 and DN2 as our kind of pro-T cell types. DN3, DN4, and DP all have pieces of the pre-T cell. As well as we sort of see our rearrangement of a light chain as the cell is transiting from double negative to double positive. So we kind of can go through that process. And we talked about our general testing the light chain, testing the heavy chain to make sure that we have a functional T-cell receptor. The key piece to think about with T cells once they have become double positives is that those T cells need to undergo selection. And when we think about T cell selection, we think about the T cells having to kind of solve 2 problems. One of them is we need to make sure that the T cells are not self-reactive. And one of them is we need to make sure the T cells bind to self-MHC. So before this step, the T cells just have a T cell receptor. They do not yet know what self-MHC is. But they are going to test against the MHC plus peptide they're seeing in the thymus. And if they have no signal, or shown here as low signal, that you can think of as very little, that means they must not even bind self-MHC. And they will fail to be positively selected and die by neglect. And that is the vast majority of T cells in the thymus. That's why that book I told you about before is Murder University. It calls the thymus Murder University. Although murder might be a little strong because we're not actively killing these cells. We're just sort of not giving them a signal to tell them to survive. So it's not really an active type of thing. However, we do have active cell death in the case of negative selection, where those cells that are getting a very high signal, either because they're binding to absolutely everything really, really strongly, or because they are binding to one thing really strongly, will actively get deleted and will undergo apoptosis to get rid of those self-reactive cells. You can see there is this other outcome that I'm not discussing right now, but we sort of have these T cells that get this low intermediate type of signal which allows them to be positively selected and survive and actually become a T cell. This is known as the Goldilocks model of thymic selection. T cell can't have too much signal. T cell can't have too little signal. T cell has to have just right, just like Goldilocks. And so this is sort of what we'd seen before. Was this slide probably should have been swapped in order with the previous one, where we saw our T cell actually doing all of its rearrangements, going through all of these steps of a process, kind of in the cortex. And now that cell is going to move to the medulla for the selection phase that we're talking about. Both positive selection, so again, the cell has to get some signal. If it gets no signal, it's going to die. It has to get at least some signal to survive and be a real T cell that's going to leave the thymus. This is how we make sure that T cells are restricted by the MHC of the thymus, because the T cell had to get some signal. If the T cell got no signal, it wouldn't work. So that's how this process results in all of our T cells being self-reactive. And we also have negative selection happening, where if our T cell is getting very strong signal in the thymus, it is going to actively be killed. And this process all relies on there being MHC plus peptide in the thymus for all of our developing T cells to test themselves against. In particular, as I mentioned, this is happening largely in the medulla, where we're doing this negative selection. And the structural cells of the medulla are that one outlier I told you about before that can present on MHC class 2. So the epithelial cells, the structural cells of the medulla, present on class 2 along with our professional antigen presenting cells. So this is how we can actually make sure our cells are testing properly against class 2. We do have class 2 available in the thymus to test our developing T cells. Hooray. When we talked about B cell development, we talked about one kind of big flaw in the B cell development plan, one issue with B cell development. That big issue with, and to be honest with you, there's a big issue with T cell development too. I actually have told you it, but haven't made it obvious yet. So we're going to come back to see what that issue is in a little bit. But the issue with B cell development was that developing B cells only have the opportunity to test themselves against self-antigens that are in the bone marrow. So if you have a tissue-specific antigen, say, oh, I don't know, insulin, or proteins in the eye, or myelin, B cells don't get a chance to test to see if they're self-reactive against that. So you can have B cells that are reactive to all of those tissue-specific antigens coming out of the bone marrow. That is, in fact, why peripheral tolerance becomes really important, because we do have these self-reactive cells coming out of the bone marrow. Things are a little different with developing T cells. Here you can see two examples of tissue-specific antigens. We're seeing a retina-specific antigen and an ovary-specific antigen here. What that means, of course, remember, every cell of your body has the same DNA, unless it's a B cell or T cell that finished VDJ recombination and threw some DNA away. But basically, the idea is every cell of your body has the same DNA, but every cell of your body might choose to transcribe different pieces. So the eye becomes an eye because it chose, I would like to make the eye proteins. And it's the only one that makes the eye proteins. except for the cells of the thymus. So cells in the medulla of the thymus also make those tissue-specific proteins. So you can see thymic medullary cells express tissue-specific proteins. So basically, the cells of the thymus medulla not only, yay, make MHC class II, they also transcribe and translate every protein that you have. That means that T cells are able to test themselves against every protein that they might find in the body. And we can delete the self-reactive T cells that react to everything in the body, including those retina-specific or ovary-specific proteins. So, yay. We actually understand how this works in that there is a protein known as AIR that basically serves as a transcription factor to turn on every protein in the body. So AIR is made by these thymic epithelial cells, particularly the medullary epithelial cells, and AIR allows those cells of the thymus to turn on every protein in the body so that we can see selection against all of those proteins in the thymus. This is actually a paper from where people had discovered AIR, and they were looking at some genes whose transcription was controlled by AIR in the thymus. And you can see that, for example, we have salivary protein, which is only made by the salivary glands, and also the thymus because of AIR. And if you get rid of AIR, then you mess up the salivary protein being made in the thymus. Or urinary protein, or oxytocin, or insulin-like growth factor, preproneuropeptide, preproinsulin. And so all of these tissue-specific proteins are made in the thymus because of AIR. AIR was first discovered in this study when scientists were looking at a group of patients who we now know don't have AIR. They have a mutation and they don't have the AIR protein. And in trying to figure out what was up with those patients, AIR was discovered. So let's think about this before I tell you about the patients. What might you imagine would happen to a patient who doesn't have AIR? By which I mean the autoimmune regulator, not like oxygen. Yeah, Michael?
It might have T cells that are self-reactive to specific tissues.
Okay, so they might have T cells that are reactive to specific tissues, right? Because those T cells aren't getting deleted in development. What would that look like to the physician? How would the physician actually diagnose them and be like, oh, something's up with you? Can you look at someone and be like, I think you have T cells reactive? Delaney? Nope, they wouldn't necessarily have low amounts of T cells. What would a physician, why would you go to the doctor and the physician would be like, I know you have, that would lead you to this? You're right, Michael, that they wouldn't have, or they'd have too many T cells. They'd have all these T cells that weren't getting deleted, and they're self-reactive. But what would that actually look like in terms of clinical outcome? Yeah? You have different tissue that's damaged. And you know what we call that? We call that some kind of autoimmune disease. But you don't have one autoimmune disease. You have multiple autoimmune diseases because you had multiple T cells that made it out of the thymus. This disease is known as APSED, which technically stands for autoimmune polyendocrinopathy, candidiasis, and ectodermal dystrophy. You do not need to know that. And in APSED patients, patients will have multiple autoimmune diseases, and it's a different combination of autoimmune diseases in each case. Because we have some other peripheral tolerance mechanisms for T cells, And so like which of T cells happen to slip through the cracks in each patient will be a little bit different. But what you can see is that 85% of APSED patients in this one study had hypoparathyroidism, had a thyroid autoimmune disease. 72% had adrenal failure. 60% of the women had ovarian failure. 18% had insulin-dependent diabetes. 14% of the men had testicular atrophy. 13% had parietal cell atrophy. 6% had hyperthyroidism. So you can see that each patient has multiple autoimmune diseases, and in fact, it is kind of a weird combo. For reasons that we don't really understand, they all have a lot of yeast infections. So they all have candidiasis, which is where the C is from there. Most of them also have some other strange things. So actually, alopecia, vitiligo are really autoimmune. That makes sense. But they all have nail dystrophy, or a large fraction of them have nail dystrophy. So they have this weird finding of their fingernails. It's not really clear what the story is on that. And so these patients, and doing genetic sequencing of these patients, allowed us to see air, and because these are all patients who don't have air. We still don't understand all of the details. People have made mice that don't have air, and they actually seem to have as severe disease as the people do. So exactly how that works, we're not sure. Though the mice have the most messed up salivary glands. They have like the worst autoimmune salivary gland thing you could ever imagine. As you can see here, different people have slightly different diseases. So all of this is really happening sort of right here as we're going into the medulla. And we left off our developing T cells as double positive cells. There is one thing that will happen after the cell is a double positive. So after the cell has made it through positive and negative selection. That is that the cell will become a single positive cell. Either a CD4 T cell or a CD8 T cell. As you'll see moving forward, when we get to the periphery, we never see double positive T cells again. Double positive T cells are only ever found in the thymus. If you see a plot that has double positive T cells, you should know that means we're in the thymus. So the cells are going to then become CD4 single positives or CD8 single positives. These are the mature T cells. And now that cell is going to leave the thymus once it has become a single positive and actually do T cell stuff when it gets signaled through its T cell receptor, like secrete cytokines, be cytotoxic, all that good stuff. You can see that here at the end as well. So after positive selection, our T cell is going to become a single positive and be basically a real grown-up T cell that's ready to leave the thymus and do whatever function it was going to do as a real grown-up T cell. There have been multiple models about how the cell decides to be a CD4 or a CD8 cell. So there actually have been 3 competing models throughout the field. This summer, there was a paper, and we discussed it on one of the podcasts, and it's pretty clear which model is the winner. And I don't want to go through all the details of it because it's super cool, but like it goes a little in the weeds. It's called the kinetic proofreading model. But basically, the cell is able to check whether it works best with CD4 or CD8. Do I get a signal with CD4? Do I get a signal with CD8? Which tells it, does my T cell receptor use class 1 or class 2? Basically, does class 1 work best for me or does class 2 work best for me? The cell is actually able to get a signal and it can tell the difference. And if class 1 works best for that T cell receptor, then the cell turns on CD8 or keeps CD8 on, turns off CD4. The cell gets a good signal from class 2, it turns off CD8, keeps on CD4, becomes a CD4 T cell. And now these cells are going to leave the thymus and head into the periphery, into different peripheral organs to respond to some antigens. So we need to think about those T cells in the periphery in a few ways. And the first thing that we need to think about with those T cells in the periphery is that just like with the B cell situation, as I mentioned, there is a little flaw in this plan. With the B cells, the flaw was, well, we don't test against every antigen. So we're going to let some self-reactive B cells out of the bone marrow because every antigen wasn't present in the bone marrow for testing. The flaw is a little bit different in T cell development. And once people actually get what this flaw is and really see this, they often will spend part of the rest of their day wondering how are we alive? This should be a disaster. This should not work. And let's see, we'll use Goldilocks. The problem here has to do with the way that selection is set up, particularly the way that positive selection is set up. So makes sense that if we have cells that are seeing antigen and getting a very high signal very strongly in the thymus, they're going to die, right? Makes sense that if we have cells that are getting no signal in the thymus, they're going to die because they don't get a nice little pat on the head. All of the T cells that leave the thymus and go to the periphery, here's a T cell that leaves the thymus and goes up to the periphery, had to be positively selected. What does it mean to be positively selected? What has to happen for you to be positively selected? Yeah. You have to react with an MHC plus peptide that you see in the thymus, right? You have to be able to get some signal from the MHC plus peptide in the thymus. What kinds of MHC plus peptide are available in the thymus? Yeah. Self. Every antigen that's in the thymus is self. So in order to become a T cell that goes into the periphery, you have to be a little bit self-reactive. If you're a lot self-reactive, you're going to get negatively selected and die. But the only thing you had to get a signal from in the thymus is self-antigen. So every single T cell that develops is a little bit self-reactive. The idea is that maybe a little bit reactive, you get a tiny amount of signal from self, but maybe you'll get a lot more signal from a foreign peptide, right? Because there's not that much possible variation in peptides with the numbers of amino acids and stuff. So you get a low signal from one of the ones that's in the thymus, you must bind to MHC, and hopefully you're going to bind really strong to a peptide that's not in the thymus and do your thing. But what that also means is that every single T cell that has come out of your thymus is weakly self-reactive. So it's sort of like your immune system has evolved to balance right on the line of having an autoimmune disease. If this went a little bit off, then we'd start really letting autoreactive T cells out of the thymus. And the idea here is that we've evolved to have a really broad immune response, the broadest possible immune response, even though it puts us at risk of autoimmune disease. One thing that you might realize here is that peripheral tolerance, the ways we get tolerance to happen in the periphery, are kind of important. if every single T cell we have that comes out of our thymus is weakly self-reactive. But you can also kind of start to see, why autoimmune diseases may be as common as they are. If you think about this evolutionarily, most autoimmune diseases tend to present in adults. We'll say like 20s, 30s, 40s. Most infectious diseases happen to kill kids who are less than 5. So we've evolved to live past 5 by having a really nice broad immune response, have enough time to survive and reproduce. If we've reproduced by the time we're 40, it doesn't really matter if we have an autoimmune disease because we've already survived and reproduced. Evolution isn't going to do anything about that. And so we've evolved this super broad immune response that puts us, like I said, right on the tightrope edge of having autoimmunity. We also oftentimes will do some experiments to help us understand some details of T cell development by looking at the periphery to see what types of T cells are present after T cell development has happened. So I'm mentioning these details about T cell responses in the periphery because I do have a couple of last T cell development related experiments to show you. But those T cell development related experiments all are measurements of T cells that you get in the periphery, kind of what was the product of T cell development. So I need to mention the periphery to you going forward. So this is what we often see for T cells in the periphery. Usually, when we think of the periphery, we think about the lymph node or the spleen. Those are the two most famous secondary lymphoid organs. And here we are looking at cells in the lymph node or the spleen and their expression of CD8 and their expression of CD4. I will also tell you that for most of the purposes of when I say the periphery, as far as I'm concerned, the lymph node and the spleen are interchangeable. So what you can see in the lymph node and in the spleen is that we have a good population of cells that are CD4 positive and CD8 negative. These are the CD4 T cells. They respond to MHC class 2. They're going to do CD4 T cell stuff. Hooray! We have a nice population of CD8 positive T cells. They are CD4 negative. They're in the periphery. They're going to do CD8 positive T cell stuff, like be cytotoxic. Hooray. I don't know why they have double positives here, but there should not actually be double positives there. You can see that it is a pretty small percentage. And just for comparison's sake, in the thymus, the double positives are the majority, like 80%. You can see that in these images, we also have a number of cells that are neither CD4 positive nor CD8 positive. What are those cells? Yeah. Those are any other kind of leukocyte. They might even be a B cell. They're basically the not T cells that are in the spleen or the thymus. Sorry, the spleen or the lymph node. And so if I were to draw one of these plots of the periphery, I would pretty much draw it like that. I wouldn't draw those double positives. I could also set up my experiment where before I did this plot, I could have gated on just T cells. So remember that CD3 is an important protein for T cell receptor signaling. I could have looked at CD3 first so that I was only examining T cells. And then I could have done this same plot. And if I did that, I would end up with a plot that looks like this, where I only have my single positive CD4s and CD8s. And I wouldn't see all of those double negative non-T cells. So this is usually what we're thinking that we're looking for with T cells in the periphery. The one on the left is ungated. The one on the right is gated on CD3. Yes? That was your question? All right. So we can, like I said, look at the outcomes of different types of mutations that should impact T cell development by looking at what we see in the periphery. And one of the first things that people have done, or people did, was that they did some experiments where they had mice that either did not have MHC class 1 or mice that did not have MHC class 2. So here we have control mice. We have mice who are missing MHC class 1. We have mice that are missing MHC class 2. And we can look at the thymus to look at which populations of thymocytes are present, as well as looking at the periphery to see which types of cells are present. So when we look in a normal thymus, we get the plot of the normal thymus. There are so many double positives. If we look at the same thing in mice that are missing MHC class 1, we find that we don't get the CD8 positive cells. So we still get these other three populations, but the CD8 positive cells are missing. If we look in the periphery, again, the CD8 positive cells are missing. Alternatively, if we used a mouse that was deficient in MHC class 2, we would see a normal plot with CD4s missing. So it would be the same idea. Now CD4s would be missing, and so on here. These types of experiments, I'm just going to draw it back so that all three of these plots match. They're all the class 1 deficient plot. These types of experiments taught us some important things about T cell development. Originally, when immunologists looked at the thymus and saw those 4 different populations, the double negatives, the double positives, and the 2 different types of single positives, they didn't know which order they happened in. They didn't know that these were the earliest cells, and then these at the end. Maybe in order to become a CD8 T cell, maybe it went like this. So we know that it actually goes like this, where you go from double negative to double positive, and then to single positive. But they didn't know that. So maybe it went like this. Like, maybe in order to have CD8 here, you had to have it here first. They didn't know. But when they did experiments like this, they found out that you could have a double positive and not have single positives. And so that said, well, single positives must come after double positives, because you still get the double positives. You must not need MHC class 1 and MHC class 2 to turn on CD4 and CD8 in the first place. You just turn them on. You don't care about class 1 and class 2. And then you use class 1 and class 2 to actually pick which single positive to be. And so these were one of the types of experiments that helped to describe this part of T cell selection. The other type of experiment that people did was an experiment using something called a TCR transgenic mouse. For time reasons, I didn't tell you about BCR transgenic mice, though there is basically a same exact thing that you could do with B cells. And I just didn't tell you about it with B cells. But we're going to talk about it a little bit with T cells. So what you do in the case of TCR transgenic mice is you take some mouse, who's just great, you just love that mouse, and you have that mouse make a T cell response. Perhaps you infect it with influenza so that you get it making a really good influenza T cell response. In this case, the mouse was of the H2K MHC type. We infected it with influenza. And then we could go in this mouse, go through its spleen, and go through all the cells of its spleen. And we could find a T cell that bound to influenza using an H2K MHC. Basically, we could find the perfect T cell out of this mouse. And we could be like, I love this T cell. This is the best T cell ever. I want to spend my life studying this T cell. What we could do is we could look at our T cell and find the T cell receptor genes. The T cell receptor alpha gene that this T cell uses, the T cell receptor beta gene that this T cell uses. There will have been some kind of VDJ recombination to make that particular beta, and some VJ recombination to make that particular alpha. You can then do some genetic engineering of an embryo to put in that pre-rearranged TCR beta and that pre-rearranged TCR alpha. In fact, every cell of the whole mouse's body will have that pre-rearranged TCR beta and pre-rearranged TCR alpha. The random mouse skin cell will have that. The random mouse skin cell does not care because it does not use the T cell receptor genes, so no one cares. But it is in the random mouse skin cell. When those T cells in this new baby mouse that you grew from your embryo, when those T cells go to the thymus and start to develop, the first thing that will need to happen is, they need to try to make a good beta chain and put it on the surface and get a signal, right? That's how development works. Except those cells won't need to do VDJ recombination to make a good beta chain. They already have one. You put it in with genetic engineering. So every single developing T cell is going to be like, whoa, look at this beta chain I found. I don't have to do any work of making a beta chain. I got a good beta chain. Yay me. And then it's going to need to test and make an alpha chain. But it isn't going to have to do any recombination because it already has an alpha chain that you put in. So that alpha chain is going to get put on the surface. And the T cell is going to be like, sweet, I'm good. And every single 100% of the T cells in this mouse are basically going to be copies of that original T cell that you took here and used as your template for your genetic engineering. So you're going to get a mouse where 100% of the T cells that develop are going to be this influenza-specific T cell that recognizes H2K thing. And so you can see that as we put that receptor in, all of those cells are double positives. They're all going to realize, hey, I respond to MHC class 1. And then if we look at the thymus, we're going to see just some double negatives, some double positives, and just the CD8 single positives. And we're going to have a mouse where 100% of its T cells will respond to influenza. This is awesome for you as an immunologist because now you basically have as many influenza-specific T cells as you want. You have like this amazing source of kind of infinity of this T cell. And you can actually do some other cool experiments to mess with T cell development as well as all sorts of things. People do these experiments all the time. You can imagine what would happen if this mouse got infected with influenza. What would happen if this mouse got infected with influenza? Yeah. It would beat up that influenza real bad. Because it had so many T cells, it would just be the most amazing responder to influenza. Kind of, but whatever. For our purposes, it would. Or for at least our purposes today, it would. What would happen if this mouse got infected with SARS-CoV-2?
It would not be good.
It would not, what do you mean by it would not be good?
It has like no T cells to
It would make no T cell response. It would die. This mouse is a really great tool for research. In different types of research facilities, we have something called specific pathogen-free facilities. We actually keep our mouse facilities clean without microbes in it. This mouse is fabulous for research purposes. So many cool experiments we can do with it. If this mouse escaped the lab and tried to run free in the world where there are pathogens, this mouse would die like that. Because there are all the pathogens besides influenza. But in the lab, in response to influenza, or just in a clean place where there are no pathogens, this mouse is in great shape and is going to be a great thing for us to use for research. I mention that because I have definitely had a lot of people get confused about that point multiple times in the past. You could make a transgenic receptor that recognizes some peptide on MHC class 1. You would get a mouse where 100% of that mouse's T cells were CD8s. Alternatively, if you made a mouse where the transgenic receptor recognized a peptide on MHC class 2, you could get only CD4 T cells to mature. And so this helps us understand how MHC is involved in T cell development. Other modifications of these experiments helped us understand some other steps of T cell development. So again, remember that in our normal thymus, we have our double negatives, our double positives, and our two single positive populations. Remember that I told you that once upon a time, immunologists knew there were these four populations, but they didn't know which one was first and which one was second and which one was third and which one was fourth. I showed you some experiments that helped us realize that the single positives had to be last. But that still doesn't tell you which of these two are first. It just tells you that those are last. Some other types of experiments helped us figure out the others. One of the ways that people did this was doing that same type of experiment with a transgenic mouse, where those cells were either responding to matched MHC or non-matched MHC. So what they're doing here is they're taking their original mouse, they're taking out the alpha and beta chains of a specific T cell that responds to a particular MHC plus peptide in that mouse, and they're putting these genes in an embryo. When they do that, they could take the original T cell from a mouse of one MHC type, like MHCA, and then when they put it into an embryo, they could put it into an embryo that has that MHC type. So the mouse has the kind of MHC that is self for these T cells. And the T cells will try to develop, and the T cells will in fact develop correctly. Because this was a class 1 MHC, we only got our CD8s. However, they could also do this experiment where they put those genes into an embryo of a different MHC type. So all of the T cells are going to develop. They're all going to try to make this receptor that you put in. They're all going to make the receptor be like, I got a great receptor. Thymus, give me a signal so that I can develop. And the thymus is going to not have any matching MHC. And so none of those T cells, when they develop, are going to be able to get a signal. All of them are going to undergo apoptosis. So you aren't going to be able to get any of your single positive cells. And in fact, you're not going to get much in the way of your double positive cells either. You get like some double positive cells and then they die. And so this sort of experiment helped them understand that double positives were second and that double negatives were first. There's also a super cool experiment that I'm not going to go through because of time reasons, where they put in either self or non-self reactive T cells to see how that changed T cell development. It was really cool. It was also a long time ago and had some weird tech problems, and so it becomes much harder to discuss than we might like. So now that our T cells have undergone T cell development, they are able to go to the periphery and make an immune response. This marks kind of a shift in class a little bit. We are finally now in the periphery. We are finally going to do something about pathogens. We have largely, through much of this semester, been putting together an immune system, and finally we get to do something. And the first thing we have to think about is what the heck I mean by the periphery. I'm going to say a little bit about it now. There will be a day later on where we talk about this again. But as you can see here, once those T cells leave the thymus, they're not going everywhere equivalently. They are often ending up in a few specific other organs. And so after either our B cells have left the bone marrow or our T cells have left the thymus, they'll go into the blood flow, which is depicted here by the heart, but they will end up then distributing through a bunch of different parts of the body. So some fraction of them are going to go to the barrier organs. This says that's about 10%. I would say that our ability to measure that is sketchy. So sure, 10%. I've actually heard it as 90% too. Sure, fine, great. And you'll see some fraction of them go to the spleen, and some fraction of them go to the lymph node. And so we are really seeing secondary lymphoid organs as a location for the majority of those cells. Those cells are able to leave the secondary lymphoid organs either through the lymphatics or through the blood and keep circulating around the body. And so this is another view of this. I really like this from another textbook, although, yes, I will admit, the numbers do not add up to 100. Don't freak out that the numbers don't add up to 100. If the cell stayed in the blood and just did circles of the body, around and around and around, those circles would take 30 minutes. And so one of those T cells could circle your whole body in the bloodstream every 30 minutes. However, as you can see, those cells are often leaving the blood to go either to mucosal tissue, spleen, or lymph nodes, and they might stay there for a few hours and then either go back to the blood or go to other tissues through the lymph. And so this is kind of what we're talking about when we talk about the periphery. When we think about an adaptive immune response, remember that one of the definitions of the adaptive immune response is that the adaptive immune response takes longer than the innate immune response. So here you can see time of a first infection, and you can see a really good antibody response and a really good T cell response, sort of at about 10 days, between 1 and 2 weeks following that infection. So there's this gap in time. People always want to see exact numbers when they look at this graph. They always remember, what day? Is it on Tuesday? What's going on? So I always put this figure from my dissertation where I did actually measure exact days. But honestly, the exact days depend on which pathogen and what the dose was and how you gave it to the mice. But there is an example of exact days if you wanted. And you can see that I had my max at, actually, I believe that was day 12 in those mice. And so when we look at this, and as we spend the next bit of time in class thinking about aspects of adaptive immune responses, one kind of question that we're often going to be thinking about in this process is, what the heck takes so long? Why did it take 12 days to get a good CD8 response in my mice in that experiment? Why is it taking 10-ish days to get a good response in this? What's the adaptive immune system doing? I mean, you did VDJ recombination before you were infected. The T cell with the perfect alpha and beta chain existed before infection. It existed way out here. And yet, it took all this time, it took 2 weeks for it to actually do something. And so a lot of what we're going to be thinking about is, what the heck is taking so long? What are all of the events that have to happen in order to get this ideal adaptive immune response? These are two different versions of sort of this timing. But what you can see is that one thing that has to happen is this problem of kind of transportation. So we're going to have some transportation. We're going to have some cells dividing or proliferating. We're going to have some cells maturing or differentiating. And we've got a bunch of different pieces that are all going to be happening here. And so this is sort of, again, just another view of this. For the T cell response, we are going to take up to 24 hours to get the antigen to the place where it needs to be. And we're going to take up to 24 hours to get the T cell and the antigen together. And then the T cell isn't even going to start doing its thing for a couple days. Similarly, we can see that with B cells, we've got some migration issues, and then we've got proliferation and differentiation issues. And so our first topic that we're going to be talking about, just a little bit, I'm going to introduce today, and then we're going to talk about in more detail on Wednesday, is this transportation problem. So if the first step is we've got to get the antigen and the T cells into the place where they need to be, let's talk about what exactly is going on with that transportation. With the antigen, things are relatively easy. Remember that we have this lymphatic system that we talked about earlier in the semester that is collecting up the interstitial fluid to bring back to the blood. Remember that water so we don't become Jabba the Hutt. And any antigens that are in that fluid can also get picked up and moved through the lymphatics and get delivered to a lymph node. So you can see these little red dots getting delivered to the lymph node here. You can also see some bacteria and other little red dots getting delivered to a lymph node on the right. There are also a number of cells. Oh, and so we have lymph nodes that are draining all sorts of different parts of the body. Each region of your body, of course, is being drained by an individual lymph node that is basically checking what is going on there. A few weeks ago, I got both my flu shot and my COVID booster, the same arm. Your arm drains to a lymph node called your axillary lymph node, which is in your armpit. Believe me, I could have circled my axillary lymph node for a few days because I could tell all of the drainage that was going on to that lymph node. And so there is sort of a specific lymph node for each different area of your body. We also have cells in the tissue. Generally, they are dendritic cells that are really good at doing phagocytosis of stuff. And basically, once they have phagocytosed something, they book it through the lymph, and they move to a lymph node. Sort of like as soon as they found something, they're like little, investigators. As soon as they find something, they head to the lymph node. And so you can see we can also have our dendritic cells carrying antigen towards the lymph node. So getting the antigen to the lymph node isn't really that hard. The lymph node isn't just all homogeneous. There are different areas within the lymph node. So we do actually not just have to take stuff to the lymph node, we have to take it to a particular location within the lymph node. You can see, for example, we have blood vessels going in and out. You saw that when we did the dissection. We also have lymphatic vessels coming in, the afferent lymphatic, and lymphatic vessels going out, the efferent lymphatic. We have areas like one of which is often referred to as the T cell zone. As you might guess, T cells are there. We have an area that's a B cell zone. As you might guess, B cells are there. We also have some really specialized blood vessels called high endothelial venules that are coming through the lymph node. High endothelial venules, like I said, are specialized blood vessels that are really good at allowing immune cells into lymph nodes. They're really nice kind of trafficking spots. For the longest time, I didn't understand the word high endothelial venule. I was like, what is high endothelial? Like, is it like on a throne? Like, what? I don't get what it means. And then I found out that what high endothelial venule means is that the cells in the venule are tall. So they're tall and skinny, so they're high up. And I thought that seemed much less fun than I was maybe hoping or expecting. Naive T cells, so T cells that have just left the thymus and are trying desperately to find their antigen. And we, as the organism, kind of hope they don't because we don't really want to have a pathogen. But those T cells are trying desperately to find their antigen. The place where they're going to be looking is the lymph node. They could either enter the lymph node through the blood vessels, particularly using the HEV, or they can come using a lymphatic vessel from a previous lymph node. This process that they use in order to do this is called trafficking. I'm going to talk about trafficking, thinking about how a T cell goes to a lymph node, which you can see here in the middle. I may also make reference to a couple of other situations. For example, if we have an activated T cell that needs to go to a site of injury, say your SARS-CoV-2 specific T cell now needs to go to the lung and kill the virus. That is also a problem of trafficking, of that T cell finding its way to the lung. When neutrophils needed to go to sites of inflammation back in the innate immune response, that was also a trafficking problem. In fact, all of these problems use the same process to be answered. So I could have done this when we did innate immunity. Instead, we're going to talk about the trafficking issues now, specifically of our naive T and B cell going to a secondary lymph node. I will point some of this out just because sometimes I've gotten some angry messages from people on social media who are like, there's no possible way that a vaccine that you get in your arm protects you against a virus that comes in your nose. What are you talking about? And the answer really has to do with both the trafficking of antigen with the dendritic cells, like I just showed you, as well as the trafficking of T cells to different locations in the body. It's all a trafficking problem. So when we think about trafficking, we can actually study leukocyte trafficking in live animals. I'm not going to go through the details of this, but we actually do have a type of microscopy where we can actually watch cells in a live mouse moving. It's called intravital microscopy. And I'm going to show you some images from intravital microscopy. But I want to just leave you with this sort of general problem that we see with trafficking to try to think this through. So we talked before about this idea of the pressure that the blood cells are under, how they're moving through the body. And we're going to look really briefly right now at actually an image, possibly, of cells moving in a live mouse. Oops, all right, share screen, this one. All right, so what I want you to notice here these are blood vessels moving blood in the mouse in real time. You can see there are a few cells outside the vessel, but you can see the general movement of those cells. When you look at, and I want you to think for a second also about the diameter of those cells related to kind of how they're moving. Yes, see, sometimes the mouse breathes. And we get things moving like that. If you look at that, does that remind you of anything? Does that make you think about anything? Yeah, Jameer. Like a river. Okay, it is, in fact, liquid flow. It's flowing real fast. Anything else it might remind you of? of things going real fast? Yeah.
Traffic.
Traffic. Think about driving on 95, right? Many lanes going super fast. And think about the fact that when you are driving in that traffic, you need to actually know, oh yeah, that's my exit, right? So there needs to be markers that tell you which location is which. Also, what would happen if you're driving down 95? I'm picking 95 because my sister came to visit me this weekend, and she drove on 95. What would happen if my sister was on 95, and she's like, oh yeah, that's the exit, brakes, while she's like in a random middle lane, like here? What would happen? Yeah. Not only would there be a backup, people would smash into her. Don't think of how dramatic that would be if she just like tried to stop. I'm going to stop now, and I'm going to turn left, right? She needed to do things like pull over into the exit lane. She needed to slow down, all sorts of things like that. We can see parallels to all of those steps in leukocyte trafficking for how exactly all of these cells figure out where they're going and say, oh, right, I'm at the lymph node now. I need to leave the blood and go into the lymph node. And we're going to see this very specific set of steps that cells do, whether they are the neutrophil going to a site of inflammation, whether they're a naive T cell going to a lymph node, whether they're an activated T cell going to a site of infection. It's the same set of steps, and we're going to see those steps on Wednesday. So I will see you guys then.